Good afternoon, class. Today we are going to learn about meiosis, an issue of cell division on human health. This lesson will be conducted by 3 teachers. Cikgu Irene Lu Leping from SMK St. Anthony Sarikei, Cikgu Tan Tuan Hung from SMK Tinggi Sarikei, and Cikgu Won Lee King from SMK St. Anthony Sarikei. The learning standard for first session are: state the meaning of meiosis, identify types of cells that undergo meiosis, and state the necessity of meiosis. What is the meaning of meiosis? Meiosis is a process of cell division that occurs in reproductive organs to produce gametes that contain half the number of chromosomes, which is haploid, of the parent cell, which is diploid. It occurs in the testes of male and ovary of female for animals and humans. These 2 diagrams show the male reproductive system and female reproductive system. These are testes. These are ovaries. So these are the place or organs where the meiosis takes place to produce gamete. What are the necessity of meiosis? Today we are going to look at the need of meiosis in the formation of gametes, gametogenesis, producing genetic variation, and maintaining diploid chromosomal numbers from generation to generation. Meiosis forms gametes through the process of gametogenesis. The gametes that produce are haploid, means that it is half the number of chromosomes of the parent cell. The process of sperm formation, we call it spermatogenesis. The process of ovum formation, we call it oogenesis. So you can see from the diagram, spermatogenesis and oogenesis, they start from the parent cell, which has 4 chromosomes for this parent cell. At the end of cell division, which is meiosis, we can see in the daughter cell that produce, the daughter cell have how many chromosomes? Yes, 2 chromosomes. Meiosis also produces genetic variation in the same species. The individual of a species have similar characteristics, but they are rarely identical. The differences between them is called variation. How meiosis produces genetic variation, we are going to learn into detail in the second session of this lesson, the process of meiosis. Genetic variation enables offspring to survive better in this ever-changing environment. Meiosis also ensures that the diploid chromosomal number of organisms that carry out sex reproduction is always maintained from one generation to the next. This is important to make sure that all the offspring's chromosomal number is definitely same with parental chromosomal number for continuation of life. So class, have you ever wondered how if mitosis happen in the formation of gamete cells? Well done. Then each gamete produced by the reproductive organs will contain a complete set of chromosomes. We look at this example. A male have a chromosomal number of 44 plus XY, female 44 plus XX. These are parent cells, which are diploid. During the process of meiosis, they produce haploid gamete, which contain half the number of chromosomes of the parent cell, 22 plus X, 22 plus Y for male, 22 plus X for female. They are gamete, haploid gamete. During the process of fertilization, when sperm fuses with ovum, the zygote is formed. The offspring that form will carry the chromosomal number 44 plus XX, 44 plus XY, 44 plus XX, and 44 plus XY. So we can see the offspring that produce have the same chromosomal number as their parent cell after fertilization. So if mitosis occurs in the formation of gamete cell, then the gamete that produced will be diploid. They are exactly same as the parent cell. So after fertilization, what is the chromosomal number of the offspring? Yes, the chromosomal number of the offspring that produce are 88 plus SSSY, 88 plus SSSY, 88 plus SSSY, 88 plus SSSY, which is double up the chromosomal number of the parent cell. But this does not occur because the meiosis occurs to produce the haploid gamete, not mitosis. So in order for the offspring to have the same chromosomal number as their parents, the cell must undergo meiosis.
Okay, students, now let us go to the second part about the meiosis. In this second part, we are going to learn about how to explain the stages of meiosis in the correct order. At the same time, you are able to draw and label the cell structure in each stage of meiosis 1, meiosis 2, and cytokinesis from the diagram later I share. Proceed to learn further about the stages of meiosis. I would like you to learn or to discuss about one important term here that is called homologous chromosome. This is human karyotype. Human karyotype shows 46 chromosomes, means our humans have 46 chromosomes, and they are all arranged in pair. Since there are 46 chromosomes, there are 23 pairs of chromosomes. Pair number 1 until pair number 22 are called autosome. Pair number 23 are called sex chromosome. Do you ever wonder why these chromosomes are arranged in pair? They are arranged according to the same size. Okay, now I enlarge one of the pair of the chromosomes. Each pair of chromosomes are called homologous chromosome. Why they are called homologous chromosome? Because one of the homologous chromosome is called paternal chromosome, another one is called maternal chromosome. Paternal means they are come from your father, maternal means they are come from your mother. So you have your father genetic and your mother genetic materials. After replication in the S phase, each homologous chromosome have their own sister chromatid, that is, they copy from their own. Okay, then the purple color and the green color chromatids are called non-identical chromatid because they come from different chromosomes. Cell undergo meiosis, that is the reproductive cells. They also will go through the cell cycle, same like mitosis, which has interphase. G1, X, G2, then go to M phase. Now M phase here is meiosis phase. Refer to meiosis phase. To produce 4 total cells. Okay, meiosis will divide 2 times. Meiosis 1 and meiosis 2. Meiosis 1 has its own phase called prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2 also have 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, let you see the overall pictures about the stages of meiosis. The cells go through prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, interphase, then prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, now let us go to the stages of meiosis in detail. We begin with meiosis 1. The first phase of meiosis 1 is prophase 1. These are the diagram. This is the diagram to show the cell in the condition of prophase 1. As you can see here, the chromatin shortens and thickens. The shorten and thickens make the chromosome become visible under the light microscope. Then you can see also homologous chromosome pair up in the process of synesis. Let the process pair up the two homologous chromosomes is called synesis to form bivalent. The meaning of bi here or B is called 2. So we have two homologous chromosomes pair up. So this structure is called bivalent or tetra. Tetra meaning here we have 4 chromatics. 1, 2, 3, 4. 4 tetra. 4 meaning tetraks. So when they are pair up and close to each other until some part of the chromatic overlap on each other, this point of overlapping is called chiasma. Okay, what is happening for the chiasma? That is crossing over. Crossing over occurs at chiasma or chiasmata for plural for exchange of genetic material between non-sister chromatids. So you can see here, after crossing over, the homologous chromosome now is different with the previous one because of the exchange of genetic material at chiasma. The purpose for crossing over is to produce variation among the same species later. Then the other events happen, that is the nuclear membrane disintegrates, the nucleus disappears. Okay, the two sides of centriole will move to the opposite pole of the cell and spindle fiber is start to form. Okay, before we proceed to the next phase, that is metaphase 1, I want you to look at this number of chromosomes in this cell. Plus, how many number of chromosomes for this cell? 1, 2, 3, 4. So these cells have 4 number of chromosomes. I want you to remember this. Later, we will discuss again about the number of chromosomes and see the difference. Okay, now we proceed to metaphase 1. After phase 1 finish, okay, we continue with the metaphase 1. The homologous chromosomes now are arranged randomly at equatorial plane. Okay, this is the equatorial plane. They are arranged randomly, means they can arrange any place they like. Okay, maybe this red go to the other side, the blue go to the other side. Okay. Sister chromatics are still tied together. These two sister chromatics are still tied together by the centromere. One chromosome from each pair of the homologous chromosome is tied to the spindle fiber from one pole cell, and its homologous is tied to the spindle fibers from the opposite pole cell. Meaning here, you see the spindle fiber is tied to the one homologous chromosome, and another spindle fiber is tied to another homologous chromosome at the centromere. After the end of the metaphase 1, now we continue with the anaphase 1. Okay, this is the picture of the cell during anaphase 1. During metaphase 1, we have mentioned that the spindle fiber attached to the one end of the centromere and another spindle fiber attached at the end of one centromere here. Okay, why the structure like this? This is because when the spindle fiber contract and shorten, it able to pull the homologous chromosome to separate from each homologous pair and be pulled to the opposite poles of the cell. Each sister chromatids moves as one unit because they are still tied together by centromere. Okay, see? There are two sister chromatids together tied by centromere and they move as one unit. This creates haploid number of chromosomes at each pole of the cell. We count again. At this pole, we count the number chromosome of the cell 1, 2, and the other one 1, 2. So here means the number of chromosomes have been reduced to half. Just now we have counted the number of chromosomes during phase 1. There are 4, but now it's turned to 2, meaning it creates the haploid number of chromosomes. After anaphase 1 ends, the cell proceeds with the cell division of meiosis with telophase 1. This is the diagram of the cell during telophase 1. As you can see here, the chromosome already arrived at the opposite poles of the cell. Each polar cells contain haploid numbers of chromosomes that are made up of one set of chromosomes only. The spindle fibers disappear, and then the nuclei will reappear. At the same time, also the nuclear membrane is formed. Telophase 1 is succeeded with the cytokinesis process, that is the cytoplasmic division. Okay, so that it can produce 2 haploid daughter cells. Both daughter cells produced are in the haploid conditions. The interphase of meiosis 1 is usually short and the DNA does not replicate. We can say that the cell have a short break. After a short phase, the cell will proceed with the second division of meiosis, that is meiosis 2. Same, meiosis 2 have 4 phases of division. We begin with phase 2. Okay, the 2 daughter cells, okay, where the sister chromatids are still joined at the centromere. The nuclear membrane, the nuclei start to disappear. Spindle fibers start to form from the centriole. Then we proceed to the metaphase 2 after the phase 2. In metaphase 2, chromosomes are arranged random on the equatorial plane. No more homologous chromosomes. Please take note this. There are no more homologous chromosomes. Just chromosomes arranged random on the equatorial plane. Each chromatid is tied to the spindle fiber at the centromere. Okay, here I show you more clear picture. This is centromere, so each spindle fiber, okay, is tied to the centromere. Metaphase 2 ends when the centromere separate. Okay, after phase 2, we proceed to anaphase 2. During anaphase 2, again the spindle fiber contract, shorten, and pull the chromatids to the opposite poles. Remember, now it's pull chromatids to the opposite poles. Sister chromatid separate. Sister chromatid separate. Okay, for clearer picture, you can see how it pulls the sister chromatid to the opposite poles. Each chromatic at this stage is known as chromosome. After anaphase 2, the cell proceeds to the telophase 2 and cytokinesis to form 4 haploid daughter cells. Okay? As you can see here, during telophase 2, chromosome arrive at the pole of the cell. Spindle fiber disappear. Nuclear membrane and nucleoli are reconstructed, means it will reappear and reform. Number of chromosomes of each daughter is the half the number of parent chromosome, which is haploid. 1, 2. See, only the number of chromosomes is 2. The parent's number of chromosomes is 4. So it's already reduced to half. Telophase 2 ends with the cytokinesis, where the plasma membrane constricts until 2 daughter cells form. So all of them are 4 haploid daughter cells. We can call them gamete because this meiosis occurs at reproductive cell to form gametes. The genetic content also different from parent cell, and also the genetic content for each gamete also are different. You can see the different genetic material at each of the daughter cells. Okay, students, we have finished discuss the stages of meiosis. Now, I would like to share with you a video in 3D animations on how meiosis occurs for your better understanding and clearer picture. This video is come from the YouTube.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of... Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during
Students, I hope this video really helped you to understand better about meiosis and how amazing meiosis to produce gametes and then produce genetic variation in the same species so that we all are different.
Hi everyone, let us begin with subtopic 6.4 with the title Issues of Cell Division on Human Health. There are 2 learning standards for the subtopic 6.4. At the end of the lesson, week 6.4.1, students are able to explain the effect of abnormal mitosis on human health. Number 1, tumor. Number 2, cancer. week 6.4.2, students are able to evaluate the effect of abnormal meiosis on Down syndrome individuals. Introduction. The cell cycle is controlled by a special control system. At each growth phase 1, synthesis phase, growth phase 2, and mitosis phase to ensure proper division of the cells. However, uncontrolled cell division sometimes can lead to the formation of tumors, which is abnormal mass of tissue. Tumor is divided into two types, which are benign tumor and malignant tumor. A benign tumor is not dangerous and can be removed surgically. A malignant tumor is also known as cancer. Cancer that is not identified at the early stage can cause damage to the organs. Finally, cause death. What are the causes make cells turn into cancer and divide uncontrollably? Cancer is caused by several factors, such as radiation, examples, X-ray, gamma rays, UV rays. Chemical substances such as tar in tobacco, carcinogens such as formaldehyde, benzene, genetic factors, and also bacteria and viruses. This will cause the cell divide continuously and develop into a tumor. Refer to your textbook, page 108, figure 6.7, the development of breast cancer. It may start with the tumor grows from a single cell. Then, cancer cells compete to get nutrients from other tissues around them. Later, the cancer cells spread through the lymph vessels and blood vessels to other parts of the body. Finally, a new tumor develops on other organs. As we can observe, some early cancers stop growing and remain within their original location. This was called benign tumors. However, many cancers invade tissues around them and spread outside the breast to other parts of the body. This is called malignant tumors or breast cancer. Summary for 6.4.1. Prevention is better than cure. Up to half of cancer cases could be prevented through healthy lifestyle choices. Let us learn more from this video.
and eliminate tanning bed use. If you must be in the sun, use sunscreen with SPF 30 or higher. If you have a family history of cancer, you may be at increased risk. Talk to your family about their medical history so your doctor can assess your risk and help you take precautions against cancer. Screening exams help detect cancer early when it's most treatable. Talk to your doctor about the appropriate screening exams for you based on your gender, age, and risk factors.
Remember to take this simple step to reduce cancer risk for you and your family. Let us continue with 6.4.2. Evaluate the effect of abnormal meiosis on Down syndrome individuals. Any abnormality during the division of meiosis can also cause genetic diseases such as Down syndrome. Photographs show 4 different karyotypes samples. First one, karyotypes of normal male. Second one, karyotype of normal female. Third one, karyotypes of Down syndrome male. Fourth, karyotypes of Down syndrome female. The normal human karyotypes contain 46 chromosomes with 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes. But individuals with Down syndrome has 47 chromosomes, which is an extra chromosome at pairs 21. There are 3 chromosome 21. This condition is known as trisomy 21. People with condition trisomy 21 has Down syndrome cannot achieve normal life. With the right support, they can. The video was shown a group of mothers learn Makaton for supporting their children communication development. Let us observe what do Down syndrome children look like. What do Down syndrome children look like? Although not all children with Down syndrome have the same features, some common features include slanted eyes, broad forehead, flattened face, short neck, stunted body growth, a slightly protruding tongue, mental retardation. How is trisomy 21 happen? It happens because the spindle fiber fail to function during anaphase 1 or anaphase 2. As a result, the chromosome fail to separate. This process is known as non-disjunction. Gametes will have an abnormal number of the chromosome, either getting extra chromosome or less chromosome. Let us have a look on this video clip that shows that is the process of non-disjunction. Okay, so from here, the video showing that there is a non-disjunction of sister chromatic in meiosis II. Summary for 6.4.2. Down syndrome cannot be cured because it is genetic disorder caused by chromosomal mutation. World Down Syndrome Day has aimed to raise public awareness about this genetic disorder, trisomy 21. Thus, let us not discriminate against persons with Down syndrome. They need our support to lead a normal and productive life.